Hey guys, so yeah, I'll tell you about BANKSY, which I've heard you guys some of you have already tried playing with, and it's basically a spatial cell typing and spatial domain segmentation tool that um, that's very scalable. So I think the way I'll describe the mathematical framework, and basically the thing is that the framework was built with scalability in mind. And so it's quite different from the two major paradigms that exist in the field, which I'll talk a bit about as well. So in spatial omics, there are two major bunch of cells. You've got their spatial locations. You want to be able to find cell types. Cell types are characterized by the average transcriptome of that cell. Oh, sorry, by the transcriptome of a cell. But cell states can be modulated. So cell states, which are perturbations on the transcriptome, can be modulated by the spatial niche that cell resides in, or said another way, by the set of spatial neighbors that cell has, right? And the other problem within sort of spatial labeling is spatial domain segmentation. So here the idea is that a cell might reside in a region of a characteristic cell type composition, or said differently, in a region of where the neighborhood gene expression signature is similar between, and the average neighborhood gene expression is similar between cells, right? So cell type composition characterizes spatial domains. And BANKSY is a method that solves both of these problems in a single framework, essentially. And before we get into BANKSY, I think it's worth just touching on the two major paradigms within the field that address these problems. And the first paradigm is a Markov random field-based methods, which sort of more generally are probabilistic graphical models. And here, so HMRFs, or Markov random fields, are 2D or arbitrary dimensional generalizations of your hidden Markov models, right? And here, there's a bunch of tools out there, and I'll talk a little bit more about what HMRFs are, but basically the tools, I think the common theme is that they don't scale to very large data sets. They can be quite complex, and the way you solve them can be complicated. So they struggle to scale to very large data sets. And the other major paradigm is graph neural networks, or deep learning in general. And there, they also struggle to scale to very large data sets, although there's this one tool called Stargate, which is, excuse me, the gate stands for graph attention autoencoder, and that method is actually able to scale to 1 million cells. So with the exception of Stargate, most of these methods are unable to scale to very large data sets. And the other issue with deep learning methods is, they can be quite sensitive to hyperparameter choice. They can be sensitive to random seed initialization. They tend to have a lot of parameters, so they can overfit data. In fact, we did a test where we tried different random seeds, and I can show you a graph if we have time at the end to see just how variable deep learning methods can be in response to just changing the random seed, which really shouldn't affect their performance very much. Okay, how did we get started thinking about BANKSY? So we were looking at Markov random fields as a method to solve the labeling problem. And essentially what MRFs or HMRFs, hidden Markov random fields, is a 2D generalization of hidden Markov models. So you have a cell that you want to give a label to. So what you can do is you can use the observed gene expression vector associated to that cell, and you can use the labels you would give to its neighbors to try to label this cell. So what HMRFs do is that they label the cell given the observed gene expression vector, but then regularize or bias that label towards to be the same label as the majority cell type label in its neighborhood, right? And you can imagine this has like a smoothing effect, like it tends to encourage nearby cells to be labeled with the same label. And as such, it can be a good tool for domain segmentation, right? Where nearby cells should be labeled with the same domain label, but not a very good cell type identification tool. There's also a slight problem with the underlying implicit assumptions within this framework. Like, one issue is that you're assuming that the expression vector of a cell is similar to the average expression vector of the majority cell type class in that cell's neighborhood. And that need not be true because, a cell can be in a region where most of its neighbors are not the same cell type as itself. But that really is a problem associated with this particular instantiation of HMRFs called the POTS model, or that uses the POTS model. There can be other models like SpiceMix uses a more complex covariate model that doesn't suffer from this assumption. But most methods use the POTS model because it's the one that's does scale somewhat, at least. It's the simplest possible model, which is, you bias the cell towards its majority neighbors. Yeah, but so yeah, that was HMRFs. We thought, okay, this is too complicated. We want to do something simpler, or can we do something simpler? And so we just said, okay, we've got each cell has a characteristic, each cell has a measured transcriptome. Can we just reduce the problem of incorporating spatial information into the clustering problem, into the labeling problem, to a previously solved problem, which is just regular graph-based clustering? And one way to do that is to just append or concatenate some proxies of the neighborhood to the features of a cell, right? So the genes are the features of a cell. You can take the average neighborhood expression of those genes, and that's if you have M genes, you've got now 2M features, right? The cell's own expression, the average neighborhood expression. You can also be a little bit fancier and say, okay, well, genes also have spatial gradients around each cell's in each cell's neighborhood. Can we also just measure those gradients and use those as features as well? So instead of M, you've got 3M features for each cell, but you're still staying within the same framework. You still just have a feature object or gene cell matrix, which you had originally in traditional single cell data analysis. So an implication of this is that all of the traditional single cell analysis packages that exist out there, your spatially informed data frame, your spatially informed data structure is still amenable to being analyzed by those tools, right? You've not created some totally different framework like HMRFs or something, right? And so we'll see towards the end of the talk, like in addition to domain segmentation and cell typing, there's really no reason why you can't do other things. For example, we used this feature object matrix to do spatially informed batch correction, and that was better than non-spatial batch correction and so on, right? So yeah, and just a word on capturing gradients. We developed this version of the Gabor filter, which we call the azimuthal Gabor filter. Essentially, instead of a planar wave modulation envelope that you would find in regular Gabor filters in image processing, the wave modulation happens in an azimuthal radial direction in polar coordinates. So it's just a nice way to capture the gradients in gene expression around cells. So okay, so before we get into using this method on real data, I think it's worth just taking a moment to talk about conceptually what doing this concatenation is doing, right? And the way to think about it is you've got a bunch of cells in your traditional own expression space. And so here I'm taking your M-dimensional space and just visualizing it as a single dimension, right? And you might have two populations of cells that are not well separated in this own expression space. But these two populations might have characteristic cell type neighborhoods or cell neighborhoods. And when you lift it to this higher dimensional space, right, where average neighborhood expression is also features, these populations might become better separated, right? And so one use case of that could be to do denoising, right? So for example, you could find that you might have that cells might get mislabeled just because the data is noisy. And one example of that is slide-seq data sets. If you're familiar with this technology, it's essentially you've got you've got these barcoded beads, and you put your sample on top and you measure things. These beads are off single cell resolution, like they're of the size of single cells, but the technology is not a single cell resolution technology because the cells need not align with the beads perfectly. So what you might find is that the measured transcriptome for each bead is maybe like 2 or 3 cells, right? It's contaminated. So what exists out there is cell type deconvolution tools, right? So robust cell type deconvolution is a really good tool that uses reference data sets. So each cell type, it uses the reference transcriptomic signature for each cell type to help deconvolve the relative proportions of cells that exist on each bead. And they showed that on the slide-seq data set, they were able to find the granule cells that were mislabeled as like the cells that were mislabeled as granule cells and the true granule cells, right? The blue is the true and orange is mislabeled, and they correlated that with the granule cell markers and so on. And so we asked, okay, well, this requires a reference data set. We have this hypothesis for how BANKSY might use average neighborhood signatures to bootstrap the denoising problem, right? And you can already see hints here that this should work conceptually, because the true granule cells are all surrounded by only fellow granule cells, whereas the incorrectly labeled granule cells are in this region where there's a bunch of other cell types. So clearly the neighborhood signatures for the blue granule cells and these orange labeled cells would be very different. So even though non-spatial clustering is calling them out as granule cells, and RCTD is able to separate them out, BANKSY should be able to use their distinct neighborhood signatures to also separate them out. And that's what we find, right? So non-spatial clustering, as expected, it labels granule cells, but also like a bunch of other cells that are not granule cells as granule cells. RCTD, as expected, does beautifully. And BANKSY is also able to bootstrap the problem in a reference data set free way and label the granule cells accurately. And it does so for a bunch of other cell types as well, right? So this was very nice to see. That was one use case, denoising data sets. Another use case is defining niche specific cell types. And this is where this is one of my favorite use cases, because I think in general in the field, people tend to identify spatial clustering with domain segmentation only. So when people think spatial clustering, they think domain segmentation, but that's not true. You can have a single cell type residing in two different niches, two different microenvironments, and its gene expression signature might be subtly different between these two microenvironments. Not so different that non-spatial clustering will be able to cluster it as two different clusters, but subtle enough that it's biologically interesting, right? So that conceptually, you can see here, maybe the blue cell type is in two different neighborhoods here, and its expression signature is slightly different. And so here we wanted to test this out, and we tried the Moorfish mouse hypothalamus data set. And you've got non-spatial clustering marks out one of the populations that it marks out is the mature oligodendrocytes. And the mature oligodendrocytes, they sit in the anterior commissure, which is like this highway of projections. And in the general hypothalamic area. So the anterior commissure is a white matter population. The general hypothalamic area is gray matter, right? And BANKSY, again, because the neighborhood compositions of these mature oligodendrocytes versus these mature oligodendrocytes is so different, BANKSY is able to separate them out into two subpopulations. So the first question you ask, okay, are there DE genes that are different between these two subpopulations? And what are those genes associated with, right? So we found that the gray matter DE genes were generally, excuse me, associated with signaling type roles, and the white matter ones, the anterior commissure ones, were related to myelination, right, as you might expect. And actually, this plot is quite nice. So this plot is what it is the own expression for the meta gene constructed out of the DE genes versus the neighborhood expression. So it's a direct real data visualization of this schematic, of this hypothetical theoretical thing, right? So you're finding that in own expression space, these two subpopulations are highly overlapping, right? The sum of these two distributions is basically a Gaussian, right? And a method cannot separate these two subpopulations out. But when you lift it to this joint neighbor own expression space, the neighborhood distributions are different enough such that like a method that sees this distribution can easily cluster these into two subpopulations, right? Okay, so the next thing is now we switch from cell typing to domain segmentation. And here the idea is that, when you have this parameter lambda, which basically when lambda is 0, the average neighborhood expression part of the matrix gets weighted to be 0, and the own expression gets weighted to be 1. So it's like a purely non-spatial clustering method, right? Own expression space is where all the cells sit. When lambda is increased all the way to 1, the opposite happens. This axis, the own expression axis, collapses, and the neighborhood expression axis is the axis that is maximally large, right? So the neighborhood expression gets multiplied by 1, and the own expression gets multiplied by 0. And there it becomes, there the method starts clustering by the average gene expression signature around each cell, by the average expression of genes in the cells around each cell. And it becomes a domain segmentation method. And in the previous slides, we were talking about this intermediate, this middle lambda regime where it becomes a spatial cell typing method, by the way. Okay, so here, for example, just now I was talking about the merfish mouse hypothalamus data set. Here we just increase lambda to a value of 0.8, which we found to be slightly more stable than a full increase to 1. So in practice, we recommend 0.8. And there we're able to reconstruct all the known regions of the hypothalamus, like the bed nuclei of the stria terminalis, the fornix, the third ventricle, and so on, right? And these regions are labeled in Moffat's paper and in various textbooks and so on. And the other thing that we tried to test the domain segmentation on was the standard benchmark data set for domain segmentation in the field, which is the dorsolateral prefrontal cortex data set, where a bunch of biologists came together and they labeled the layers in the cortex. And we tested a bunch of methods, and there are actually 12 slices. And this is the most popular slice. I think it's the third patient, if I'm not wrong, but it's the most popular slice that everyone likes to show. But there are 12 slices, and you run the method on all 12 slices, and you report the mean and median of the ARI performance across all slices. So we found that BANKSY does just as well as other methods, although the variability, so both GraphST and Stargate are both deep learning methods, and there does tend to be a lot of variability in performance for some of these other methods, whereas BANKSY being mathematically a very simple framework that is then just leveraging lead-in clustering, right? It's a much more stable method. And so it does as well as the other methods, but its distributions are much tighter. Okay, so yeah, so that's the conceptual part of things. In terms of usability, we've tried to make the method as usable as possible. So like we just tried to do all three things. Like we have a bioconductor version, which is the default version on the main BANKSY page. We have a Python Scanpy version, and we've got a Seurat wrappers Seurat version. So you can use BANKSY directly on Seurat objects as well. And all of this is documented on the GitHub, and you can read through it. You can also go to Rahul Sathija's website and go to their spatial omics page, which is, I guess, this guy. And they also mention that, hey, BANKSY does quite well on domain segmentation. They don't mention the cell typing part of it. I really hope the field like gets enthusiastic about spatial cell typing as well. But it's much easier to benchmark, I think, domain segmentation, which is why it's like the major paradigm. But I think in time, people will talk a lot more about niche specific cell types and so on. Oh yeah, and the other use case was the spatial batch correction. So remember I was saying how like basically the output of BANKSY is this spatially informed gene cell matrix. I do this in air quotes because it's not genes, it's features now, feature cell matrix, right? And you can just feed it into whatever single cell tool you want. So in particular, for example, Harmony, right? You can take BANKSY, you can take your data, you've got your 12 slices, run BANKSY on it. Now you have your 12 spatially informed gene cell matrices concatenated or whatever, and feed it into Harmony, right? And and then you just do clustering, right? And what you find is that the performance is much better, right? So spatial batch correction. We call it spatial batch correction because you're doing batch correction on something that's spatially informed, right? This was inspired by actually a vignette that BaseSpace had, where they did Harmony on non-spatial data first, got the batch corrected versions of the non-spatial data, then they just ran BaseSpace on it, and they found that improved performance for them, right? So we just thought, hey, why not try this? Okay, so and the other part is scalability. Like I said, this is probably, okay, I mean, the method has to work well first, but scalability is like a close second in terms of how important I think it is, because like BGI has like this method that, you know, measures millions of cells, and all the data set sizes are getting larger and larger, and you need a method that can handle very large data sets on reasonably sized compute, right? And so BANKSY like on a laptop, like on my MacBook Air, I can usually go to 100,000, 200,000 cells without problems. And if I want to go to like 1 million cells, like a small Amazon EC2 or Google Cloud instances is good enough, like 128 gig RAM instance, which is like nothing, it's a CPU instance compared to like using GPUs or whatever, which are way more expensive. And so you know, and the reason why BANKSY scales, right, with non-spatial is because it's the same thing, right? It's just you're doing two deterministic steps. You're doing a neighborhood average computation and an azimuthal Gabor filter computation, which are both relatively light computations. You do that, you take a bit of extra time, and now you have a feature object matrix. You do PCA, get it down to some reasonable number of PCs, and then you do lead-in clustering or Louvain or K-means, right? So it's going to scale with the state of the art of graph-based clustering methods or whatever clustering method that you have. And I think there's a tool called BP cells, which allows you to go up to like 4 or 5 million cells. We've not implemented this, but we're looking into it. And I think, yeah, just having BANKSY work with BP cells for the clustering step would easily make it scale to, like 5, 10 million cells. So this is nice as well. One thing that we found was the authors from a university in Israel, they emailed us and they were like, hey, we've been using BANKSY to analyze MRI data. So here are the features they said were layer thickness within the cortex of the brain. So you drill a hole in the brain and, like you look at the thickness of layers. We don't actually drill the hole, but, and so those are the features. Layer thickness is the features, and you can just use BANKSY in domain segmentation mode. And they found that it does better than the other tools that were available to them in segmenting out the regions of the brain. So it was nice to see that, like algorithmically, there's no reason to just use this for spatial omics or genomics data analysis. Anything in the spatial domain, in geospatial and MRI data, whatever, you can use the method. So yeah, I think I'll stop there. There's a bunch of other stuff. Maybe the discussions and the Q&A, we can discuss other things. But I would like to acknowledge my co-first author here, Nigel, and Joseph, who actually was the software engineer that did a lot of the code. Like initially when I wrote the code, I'm not a software engineer. I wrote it in one of the ugliest possible ways you could imagine. And Joseph comes in and he's like sweet, and he's like, hey, let's do this properly. And I think you guys must have noticed, those of you who've seen the code, it's very beautifully written. That's all Joseph. Sham, Kokhao, and Huiguan were the PIs on this project, and just incredible PIs. And yeah, and the other team members as well, and funding from A-Star.